everybody! In this video I'm going to be talking to you about the human immune response, and I just thought I would start off with this beautiful image. So this is a scanning electron micrograph, and it is a false colour, so obviously they aren't this colour, but this is a macrophage. So this is a type of white blood cell that travels around your body and sends out its long strands of cytoplasm like lassos. They're called pseudopodia. And when it reaches out, it can smell the bacteria, and it travels around looking for bacteria. It can smell them. It uses a process called chemotaxis, so it can travel towards bacteria using the chemical gradients of the toxins and the waste products they give off. So it moves around the body, and then it sends out these lassos, and it grabs hold of the bacteria. We can see here it's grabbed hold of some rod-shaped bacteria. We don't know what it is. It could be E. coli, these rod-shaped bacteria. We call rod-shaped bacteria bacillus. And it's going to draw them into its body. When it draws them into its body, it's going to engulf them and it's going to digest them. So inside the body of this macrophage, there are hydrolytic enzymes. There are digestive enzymes that are going to digest down the bacteria and break it down into harmless waste products that the macrophage might spit out as pus, might use some of them, or it might present some of the bacterial antigens on its surface. And when it does that, it's triggering other parts of the immune system to kind of wake up and be like, oh my goodness, we've been infected with something, we need to do something, and then start the next stage of the immune response. So these are really amazing cells that you've got millions and millions and millions of in your body right now, roaming around like little sheriffs trying to protect you from any infection from bacteria or viruses or fungi or protists. Perhaps at this point we should answer a simple but perhaps overlooked question of how these organisms actually cause us harm. So they can cause us harm in a whole bunch of different ways, or they can cause disease in a whole bunch of different ways. But just on a really basic level, to summarize it, bacteria can rapidly reproduce inside your tissues. And when they reproduce, they can often produce and release toxins that can damage your cells. Viruses replicate rapidly inside your cells, and they kill or damage the cells in the process of replicating. So you already know that the cells of your immune system are called white blood cells. White blood cells are produced in your bone marrow, and they can travel around your body in both your bloodstream, your tissue fluid, and your lymphatic system. So the white blood cells of your immune system will attack pathogens in one of three ways. Through phagocytosis, which we've already talked about a little bit, but we'll go into a bit more detail about in a minute. Let's just take a look at that word, phagocytosis, for a second. This word, phago, means eating. So this prefix phago, eating. Cyto, cell. So this word just means cell, eating. The second is antibody production and antitoxin production, and we'll talk about each of those in turn. So in phagocytosis, white blood cells, called phagocytes, engulf pathogens and then digest them using enzymes. So the phagocytes come along to where the bacteria is. They are attracted to the bacteria, they bind onto the bacteria, they engulf that bacteria into their body, and then they fuse that bacteria with a little sac we call a vesicle that contains loads of digestive enzymes, and those enzymes break down the pathogen. Antibodies are proteins. They are proteins that are produced by white blood cells called lymphocytes. And these proteins bind onto and can destroy pathogens. Antitoxins are basically also antibodies. They are also proteins that bind to, and instead of binding to the pathogen itself, they bind to toxins and they neutralize that toxin. So for instance, many bacteria produce toxins. For instance, the cholera toxin is a toxin that disrupts the lining of your small intestine. It's what gives you diarrhea. The Botox toxin disrupts your nerve cells and causes you to be paralyzed. So antitoxins are proteins that bind onto toxins, and toxins are usually also protein-based. They're usually tiny, tiny little molecules, and then the antitoxin binds onto them and neutralizes them. So 3 ways: phagocytosis, antibody production, antitoxin production. Phagocytosis by phagocytes, antibodies and antitoxins by lymphocytes. So let's talk in a little bit more detail about antibody production. All cells have antigens on their surface. You have antigens on all the cells in your body, and those antigens are specific. They have a specific shape that your immune system recognizes. So the antigens in your body are recognized by your immune system, and your immune system ignores them. This is something that happens when you're tiny, before you're born, and when you're a tiny, tiny baby, where your immune system is being trained to recognize self-antigens so that your immune system doesn't attack and destroy your own cells. There are conditions which are called autoimmune diseases. People who have autoimmune diseases, their immune system hasn't been trained really properly, and so it starts attacking and killing its own cells. So, in general, in biology, when we think about antigens, we think about how they can cause an immune response. So an antigen is a small, usually protein-based molecule that can trigger an immune response in your body. So when a pathogen enters your body, when it gets into your bloodstream or your tissue fluid, when it gets beneath your skin, your immune system, the cells of your immune system are able to recognize that as a foreign antigen, and it triggers an immune response. So when your white blood cells recognize this, you've got all these different lymphocytes that are capable of each lymphocyte, each of these cells, and the specific types of lymphocytes that are able to make antibodies are called B cells. So I'm going to be using this term B cells from now on. Okay, so they're a type of lymphocyte. So each of these B cells is capable of making a specific shaped antibody. Each one makes a different one, but you produce many millions and millions of these B cells, each one capable of making a different shape. So your body, your immune system, is capable of making many millions of different shapes of antibodies. So when you're infected with a particular pathogen, each one of these has a go. Ooh, is my antibody the right shape? Is my antibody the right shape? Is my antibody the right shape? Until we find the one whose antibody is the right shape. When that B cell is identified, that is able to make the correct shaped antibody that is complementary to the antigen on the surface of the pathogen and will bind on to the surface of the pathogen, that B cell starts to rapidly replicate by mitosis and starts to release millions and millions and millions of these antibodies. These antibodies perform a series of functions. One of the functions is to attract phagocytes to perform phagocytosis, and the other is to break holes in the cell wall of the pathogen, causing it to break open and die. So these are the 2 key functions of the antibodies. And we can see here how the antibodies bind on to the antigens on the surface of the pathogen when they have the correct complementary shape. In addition to producing B cells which make antibodies that are really important for binding onto antigens, lymphocytes are also capable of turning into cells called memory cells. Memory cells remain in the bloodstream in high numbers after infection. And basically, their job is to remember the correct shape of antibody. So they retain that information. Oh, this was the correct shape for that particular pathogen. And they initiate a rapid response if you get infected again. This is called your secondary immune response. So the first time that you're infected with a pathogen, this is called your primary immune response. So in the first time, your body's like, what is this? I don't know what this is. Do you know what this is? No, I don't know what this is. And so you go into a bit of a panic mode. You have to spend all this time trying to find the correct antibody. And then once you've found the correct antibody, you've got to undergo mitosis. And then you've got to start producing all these antibodies. It takes a lot of time. That's why you get sick. You get sick because there is a time lag between identifying pathogen, or a time lag between the pathogen entering your body, you being able to identify it and then produce the correct antibodies in large enough numbers to basically kill it so that it goes away. So that's really why you get sick. So these memory cells essentially provide you with immunity against a particular disease. It enables you to destroy the pathogens before developing the symptoms of the disease. This is really important. Some students mistakenly say it provides you with immunity from the pathogen. Memory cells don't stop the pathogen getting into your body. They just stop the pathogens being able to invade, being able to proliferate, being able to, there to be so many high numbers of pathogens they can cause harm. They prevent you from getting the disease. They don't prevent you from getting the pathogen. They just kill the pathogen quickly once it's got in so it doesn't replicate. So let's take a minute to take a look at this graph looking at the primary versus the secondary immune response. So on the y-axis, we've got antibody concentration. This is just in arbitrary units, so you produce large numbers of antibodies in your bloodstream. And on the x-axis, we've got time. So let's have a look here. This bit here is your primary immune response. So on day 0, you became infected with the pathogen, your first exposure to the antigen. So you became infected somehow with a particular pathogen. Now, for quite a long time, for 7 days basically, nothing happens. It looks like nothing's happening. In all that time, you haven't yet started to mass produce your antibodies. In that time, you're doing a little bit of phagocytosis. You're trying to identify the correct B cell, but then you do. And as soon as you do, you start undergoing rapid mitosis and producing lots and lots of antibodies. Remember, antibodies are proteins, so this process is called protein synthesis, and the numbers of antibodies in your bloodstream rises up, and then you start to get a hold on the infection, you start to feel a little bit better, and your antibody numbers drop. Remember, also in this time, you've also started to produce memory cells, so they're also present in your bloodstream. They don't drop back to 0, but they drop pretty low. Then, on what do you reckon this is, like day 29, you have your second exposure to the antigen. You get infected with exactly the same pathogen again. Look how different the response is. Unlike the first time, there's no 7-day time delay. There's a really short time delay, a day, 2 days maximum. And then let's have a look at the curve. The curve is much steeper, telling us we are producing antibodies at a faster rate, and the line, the peak, is much, much higher, telling us in a shorter period of time we produce more antibodies. And then after the peak, we see a drop again, but it does not drop as low as it did the first time. It remains very high in the bloodstream. So we can see how the primary and secondary immune response are very different, and how the secondary immune response protects us from the pathogen, and so gives us immunity from a particular disease. This is a really important graph. Just take a minute to read through this information here. Okay, so well done for listening to that. There was lots of key information there, and I will speak to you soon.